Good afternoon, students. Am I audible?
Good afternoon, ma'am.
Okay, so that means I'm audible, right? So, okay, almost all students have joined. So, I think we can start the class, right? So, we are done with the first module, and today we are going to start the next module, and which is a very important one because this module we will discuss about how infection takes place, what are the different steps, how the pathogens they get entry into the host, what are the different ways of a penetration, right? And then how they establish contact with the pathogen, how they enter, right? So all those, and what happens after the infection, right? So a lot of things, and then how after infection, then they how they exit, and then how they survive in an extreme condition, and then how they disperse of others. So reproduction, dispersal, exit, everything is in this module that we are going to discuss. So last module we ended with the history. I told you that yes, there are certain landmarks that are important to remember. Not all history. I don't want you to mug up the things that go, but at least few, at least notable contribution for 3 to 4 people in the history that just try to go through that, right? So we'll begin with the next today's topic, and here we are going to discuss. We have discussed a lot of terms in a plant pathology, right? In the beginning, I told you that there are a lot of terms, common terms that we are going to use, like pathogen, pathogenesis, disease cycle, all those terms, right? So, what is pathogenesis or what is disease cycle or an infection cycle? They come together in one basic umbrella, right? One common umbrella. So, a series of events that occur in a succession during a pathogenic relationship of a pathogen and a host that lead to disease. So what does this mean? This means number of events starting from entry of a pathogen and how it gains entry to the symptom development, right? What are the changes that are being introduced by the pathogen in a plant? All those events, they which leads to the disease at the end, right? The changed or a changed metabolic it could change a lot of things in the plant, right? It could affect the photosynthesis, it could affect other metabolic processes and other things that could lead to some internal symptoms and some external symptoms also, right? And at the end, why we are concerned, we are talking about the losses. Fruits may be rotten due to the fungus, fruits may be smaller in size or they are totally destroyed, which we are not able to consume, right? So all those events, how all those events takes place in a proper way, there is a stepwise procedure for that, right? So study of a disease cycle also generate information about how there's a source of perpetuation of a pathogen, how it perpetuates, how it spreads, then how further at the end of the day, when we are talking about diseases, why we are studying, how we want to or how we can formulate certain management practices. Why we are studying disease? We are studying disease so that we can come up with something or some steps where we can do something for the management of the disease, right? That is why we are studying disease. Otherwise there is no use. If you are not able to manage a particular disease or control particular disease, so there is no use of studying that particular disease, right? So a lot of terms such as pathogenesis and parasite, we know parasite. Mm, See, pathogens can be parasites also, and not all parasites are pathogen. It could be that way also. So parasite, we know parasite which take benefit from the host, which steal its foods and it affect that, it's a parasite. Pathogen to also somewhat they do these things only, right? They're going to affect, they are using host for their application, they are going to affect the normal mechanism in the host, and they are going to get benefited there, right? So pathogenicity means ability of a parasite or a pathogen to interfere with one or more essential function of the plant. That is the pathogenicity or ability of the plant. That means it is able to interfere with one or more essential function of the plant, right? Oh, so pathogens life cycle, or we call it as a disease cycle also, it follows events which are like a disease cycle. Events such as, we call it a disease cycle. Basically we'll study in detail what is disease cycle. So how do pathogens it survive, how it survive when it is outside the host. For example, plants are not growing always, right? There's a season, annual plant hai, aapke perennial plants, okay, in a perennial plants we can see that, or some plants which trees such as fruit trees, so there they are, trees are always there, right? So but what about the annual plants or plants, or just their cycles, or the plants they are just harvested within a 6 month or maybe a shorter period of a time, right? So how do pathogens they survive? Then how over summering or overwintering, those stages, how they pass out those stages, how they are disseminated, then how they are being disseminated to the plants again, how to the far places, how they are carried to the other places also, how they penetrate the host, how they go inside, or how they gain entry into the host, because it is not just very simple that they are just getting entry by any means. No, maybe through wounds, maybe through other natural openings in the plants that they are using to get entry. Then viruses, we know they get entry to the vectors, right? So there are different means of getting penetration the plant also. Then invasion, how they invade the plant further, how they go inside the plant, what different structures they form. Reproduction, how they multiply, and then dissemination and survival, how they disseminate and then how they further survive. So this is something about a disease cycle or a pathogen's life cycle, which contain all these events together, right? Then again, there are certain points such as primary disease cycle or a secondary disease cycle, or a monocyclic diseases or a polycyclic diseases. So as the name suggests, monocyclic diseases means they just complete one cycle in one particular plant. That's all, one cycle. So that means they have a primary inoculum. I'll discuss the inoculum primary and all those things. Also, they have a primary inoculum. It get disseminated. It cause a primary infection. Then there's an overwintering stage, and this cycle goes on. So they have a single cycle in a particular season. Then there are secondary disease cycle, or we call them as a polycyclic diseases also. So what does this mean? This means those pathogens which completes many life or a disease cycles in a one season. So more than one cycle, that are called polycyclic. So they in polycyclic diseases there is a primary infection, then there is a secondary infection. So from a over summering or overwintering, they'll get primary infection. That means one cycle in one season, and this primary infection, whatever will be the product of the primary infection, will lead to the secondary infection further, right? So again, they'll be disseminated and reproduction. So that means completes many life or a disease cycles in one season, more than one. Those are called your polycyclic diseases, right? So, and then we have a survival of the pathogen. So that means how they survive. So there are different terms. Inoculum, I have told you earlier also that any part of the plant or of the pathogen that can cause disease is called an inoculum, right? Spores ho sakte hain, fungal spores, and they are called inoculum. So primary inoculum means which causes a primary infection. First infection could be some kind of a sclerotia or a mycelium or a spores which are left from the previous seasons. Wintering stages में जो रह गया था उससे जो first infection initiate होगा plant में, that is called primary inoculum. Secondary inoculum is derived from the primary inoculum. Could be some secondary infection, Conadia या Iridospores हो सकते हैं, या maybe zoospores हो सकते हैं. So that arise from the secondary infection. Inoculum density means number of infective propagules. How many spores were there? Were spores enough to cause a particular infection or to initiate another cycle? So that is called a spore density, right? So primary inoculum lives dormant in the winter or summer, and it causes original infections, right? Maybe in the spring or autumn. Secondary inoculum is that which is produced from the primary infection. So these terms, just remember what is primary infection or primary inoculum, what is secondary inoculum, what are monocyclic diseases, what are polycyclic diseases, right? So again, here types of inoculum, like we have a primary inoculum which is from the over seasoning stage. It causes a primary infection, and from primary infection we get a secondary inoculum. So secondary inoculum kya hai? Whatever spores are being produced from the primary infection, they are counted as a secondary inoculum. Again, this will lead to the secondary infections, as it could be repeated many times. So basically we have a primary inoculum and secondary inoculum, two types of inoculum. Then sources of inoculum. From where are we getting this inoculum? It could be, as I told you, infected host as a reservoir of inoculum. some parts of the plant which are remaining in the field. Then saprophytic survival outside the host. Fungus may survive on some other plant as a it act as a to obtain its nutrition from other plants, so they can act as a source of infection. Or there could be dormant spores or any other structure on the host or outside the host. So fungal spores, or they survive through the they remain dormant in a unfavorable weather, right? So whenever they find the favorable conditions, they are going to germinate and they are going to cause the disease again, right? So these are the sources of inoculum. Then host range of the pathogen is another term. Host, we all know any animal or a plant which is infected or invaded by any pathogen or a parasite, right? So, for example, we'll say the cucurbitaceae plants, the cucumber mosaic virus host range include cucurbitaceae plants. So, plants from this family are the hosts of the plant cucumber mosaic virus. So, host range, right? It could be some other family also. So, host range का मतलब क्या हुआ? Number of the plants or the plants of a particular group which is infected by a particular pathogen. And this is important here. Not all plants are host of all the pathogens, right? Pathogens can be specific in their host, right? And there are a number of tree viruses or tree diseases which are not causing infection in the vegetables or other plants. So it is not important that all pathogen affect all kind of a host. Pathogens are host specific, right? So host, a plant or animal which is invaded by a parasite and from which parasite obtains its nutrition. Host range means various kind of a host plants that can be attacked by a single pathogen. So when we talk about the host range of a fungi or a host range of a particular group of a fungi, so that means we need to mention the number of the plants that particular pathogen is affecting, right? So it could vary. For example, host range of viruses, it varies sometimes, right? It infects the ornamental plants also. It infects the vegetable plants. So cucumber mosaic virus, for example, a very common example. We find cucumber mosaic virus in the ornamental plants. We find cucumber mosaic virus in the your vegetable plants. We find cucumber mosaic virus in some other fruit crops also. So that means its host range is wide. It is infecting number of the plants, right? So this is how we define host range. For example, there's a brome mosaic virus which infect grasses such as barley, but brome mosaic virus is not found in legumes. So that means legumes are not host of this particular virus. So a plant species that does not show disease when infected with a pathogen is referred as a non-host plant species of that particular pathogen, right? So remember these words. What are the hosts of a particular pathogen? What are the non-hosts of a particular pathogen, right? So organisms that do not cause disease on any plant species, such as some saprophytic bacterial species, Pseudomonas, they are referred to as a non-pathogen. So definitely, those not all fungi they infect plants or a human, right? So those which do not infect, they are called as non-pathogens. Those which infect, they are called as pathogens. So these are the definitions of the terms that we are going to use right here. This I guess I have talked this about this very lot of time, like a disease triangle, right? So for any disease to take place, this condition is required: suitable environment, virulent pathogen, susceptible host, and a time period for which they are together and then they cause disease, right? So conditions for a disease: host should be susceptible, pathogen should be virulent, and environment should be favorable. So this you all know, and we are not going to discuss this in detail. This is something new, right? We talk about disease triangle many times, but there is another term which we call as a disease tetrahedron, which is being given by a lot of plant pathologists. So what does this mean and how they came up with this idea? Basically, they said that it is not only the disease triangle, but it is the disease tetrahedron also, where we put environment, host, pathogen, and then human activity. A lot of human activities lead to the disease. A lot of human activities in the field are responsible for a disease to transmit, and there's a in between there's a time period also. They say that it is not only host, environment, and a pathogen, but it is also due to some human activities due to which diseases are transmitted. How? If you're not we do a lot of activities in the field, like activities in the sense that there are a lot of steps. You do weeding, you do pruning also, sometimes pruning is required, thinning of the plants are required because they need space. So during these activities, when we are using the similar kind of equipments, we can unknowingly transmit the pathogen from one plant to the other plant. For viruses, it is very important. When we are doing pruning and we are using same chettiars or scissors to prune all the trees, so there is a possibility that from one infected tree we are transmitting the virus. Pruning is a very common phenomena in a palm fruits, apple trees. So they, after every season, they require pruning, right? So if different apple, there are hundreds of apple tree in an orchard, right? So if we are using the same shettias for pruning our trees, so definitely we are transmitting viruses unknowingly by doing this, right? So this means they want to say that it's not only the host environment and the pathogen, but also humans, they play a very important role. So they gave the term disease tetrahedron for this particular, right? So then we have a dispersal, right? Dispersal mean there are two terms here. One term is a distribution and another term is a dissemination. And these two terms, they seems to be same, but they have a different meaning. So what does distribution mean? Distribution means spread of pathogen within the geographical area where they were once established. एक पर्टिकुलर एरिया में जहाँ पे वो पहले ग्रो हो रहे थे किसी क्रॉप में, they were found in that particular crop and they were already in that area. Dissemination means pathogen is introduced into the new area where they were not found earlier. So how dissemination can be done? Dissemination can be done through the seeds. Dissemination can be done through the vegetables which are being transported to some other part or imported or exported to some other part, right? So dissemination means we are introducing pathogen to the new area. Distribution means in the similar area they are being circulated. Then they are being transmitted, how they are causing diseases to the plant. Transmission could be, again, it could be through the direct, through the soil also, through the seed also. There are a number of ways of a transmission. Spores, they remain in the soil also. Seeds, if they are infected by fungal or a viral pathogen, they are there. So whenever these seeds we are using it, we are transmitting the pathogen, right? Then we have an indirect or a passive dispersal also, could be through air, such as it is called anemochory. It is through the air. So powdery and downy mildews, rust and leaf spots and blights, they can be transmitted through the airs. How? Because they form a dry fungal spores on the leaf surfaces. And these spores can be, if there's air in a particular area or there's a lot of wind velocity is more, so these spores can be definitely taken away to the healthier plants or other parts of the plants also, or other areas also, right? Then hydrochory could be through the water. Then Plasmodiphora brassica causing club root, and there are certain examples which could be transmitted to the water. Zoochory means insect vectors and the nematodes. So they are transmitted by the insect vectors and the nematodes. And there's another term which is called anthropochory. So this term means dispersal by inadvertent dispersal, means unknowing dispersal. We don't know, maybe through the humans or through animals, could be any other way. Just they are not the established way of a transmission, but it is happening. Then some pathogen may also use their own force to reach the host, such as larvae. Larvae can move, right? Nematode larvae and zoospores can move through the waters, and some bacteria, so they can move on their own. They can reach the healthy plants and they can get transmitted. Then we have an infection. So after all these things are happening, then there is an infection. There are certain terms such as inoculation potential, and then infection. So what is inoculation? It is easy, right? The process by which a pathogen come in contact with the host is called inoculations. So we do inoculation artificially also sometimes to check the symptoms, and it is being done in the field also, right? So how pathogen is coming in contact with the host, that is called inoculation. Then inoculum potential. So that means the function of the number of infected propagules. That means how much inoculum potential means whether this inoculum which has come in contact with the pathogen has capability to cause the disease or not. So उसमें कितने spores है, उसमें how much spore density is there. So it determine their pathogenic capacity also, right? Inoculum capacity also, that whether it is able to cause the disease or not. Potential, कितना potential है उसमें. Then infection, establishment of organic relationship of the pathogen with the susceptible cells of the host. How they established, relationship with the host, with the cells of the host. That process is called as a infection, right? So incubation period, we know that, right? Incubation period is basically when the pathogen it penetrates the host till the time when the first symptom appears. So that period is called an incubation period, and the incubation period is a different for a different kind of a host. Even in fungi, also different fungi incubation period may differ from 1 week, 3 days, as early as a 1 to 3 days to 1 week or a 15 days also. So that is the incubation period. Invasion means how it is spreading in the host, how it has invaded host. So एक बार वो host के अंदर चला गया तो अंदर कैसे वो move कर रहा है, how it is invading different cells. It is forming infection bags, it is forming hostorias, and it is invading the different cells. Like in fungi, they form hostoria. So how it is doing that? Then predisposition means means the action of set of environments prior to penetration and infection, which makes the plant vulnerable to attack by the pathogen. So there are a lot of sometimes predisposition factors also. Could be the adverse environmental conditions, also could be the wounds by some vectors or some other pathogens, or some other due to some other reasons also. That means plant more vulnerable to the pathogen. So what are those factors? They also play important role here. Okay, then penetration means the process by which pathogens enter its host. Again, it is divided into pre-penetration process, penetration, and a post-penetration process. So there are 3 different steps. Pre-penetration, what are the changes which takes place in that time? Then penetration, and what are the post-penetration processes, what happens after the penetration is being done. So pre-penetration process means it again differ in different host pathogen combinations. It means attachment of the pathogen to the host. Spore germination in fungi is a pre-penetration process, that before they penetrate spores, they need to germinate, right? So suitable weather or some moisture is required so that they'll germinate, right? Or bacteria, they are going to multiply. Or in case of a nematode, they are going to hatch, right? Nematodes, they are going to hatch. There are different stages in the nematodes also, right? So one particular stage infects the plant. So these are the pre-penetration process: hatching of the nematode eggs, multiplication in the bacteria, and spore germination in the fungi, right? And then perception of the host surface, how they are going to percept that particular host surface, how they are going to reach that, how they are going to affect that, right? Then pre-penetration process, first is the attachment of pathogen to the host. So pathogens like viruses, phytoplasma, viroids, or they can directly placed inside the host by different it could be by the different pathogen vectors also, because vectors, they are doing that and they are putting in directly. Almost all fungi, bacteria, parasitic plants, nematodes, they first come in contact with the host surface, and then they should attach to the external surface. Now this is done through some kind of adhesive material or propagule surface, which consists of a water soluble polysaccharides, glycoproteins, lipids, or some other kind of the material, right? So presence of a moisture on the host surface, it helps in the attachment. So that means a moisture should be present. That is the prerequisite. Or see, exact mechanism of a spore adhesion is not known. However, they are saying that there is specific interaction of the spores with the host surface by lectins, ionic interactions, or could be some kind of a hydrophobic contact with the plant cuticle. So these leads to interaction with the plant. And then studies, they have shown that many proteins of the fungal cell wall also play important role in adhesion of the fungi as well as host surface perception by the fungus. So there are different proteins also which play these kind of roles here. Then we have penetration process in the fungal pathogens. The first spores, they are going to germinate. Resting spores होते हैं आपके. Then some spores, they germinate immediately, like Conidia and ascospores, they germinate directly. They don't wait. They can germinate directly. Other need some resting period for their oospore. So some time is required by some spores, right? So spore of a different fungi germinate according to the nature of the spores and the weather condition or environmental conditions which are present. Resting spores कौन से होते हैं? आपके asexual spores होते हैं. Resting spores can be spore created by fungi which is thickly encysted, has a thick cell wall, and they survive through stressful conditions such as drought, right? Resting, they rest a particular unfavorable time and then they germinate when the time is weather conditions are favorable. Then sexual spores such as telospores or noospores होते हैं. Propagative spores such as conidia and zoospores होते हैं, right? The spores may germinate soon after formation, or they need a certain dormancy time. But the moisture is one of the important factor, followed by the temperature. So suitable temperature and the presence of the moisture is required for the germination of the any kind of the spores to cause infection, right? So the infection structures here, this figure shows the infection structures. We have a germ tube here, then there's a aprosporium, it is being formed, then there's an infection hyphae, hostorias are being formed, then there's an infection cushions and the rhizomorphs. So rhizomorphs are basically thread-like structures, right? They are made up of a parallel hyphae and a branched tubular filaments that make up the body of the typical fungus. So rhizomorphs can act as an absorption and translation organs of the nutrients also. So basically, first the germ tube is formed by the germ spores, then it forms the aprosporium also, and infection peg and hostoria. So these hostoria, they enter different kinds of the cells further, right? So again, the spore germination and perception of the host surface. So that means stimulation by contact with the host surface, hydration and absorption of a low molecular weight ionic material from the host surface, and also how much nutrients are available, it plays important role in a spore germination. So stimulations which are received by spores, it mobilizes their food reserves such as lipid polysaccharides, right, or a spore contents, and then they direct them to cell membrane and cell wall for formation of the spore tubes and its extension. So this is how they are the perception of the host surface by the spores. Then okay, the germ tube is a specialized structure which often grows short distances before it differentiates into aprosporium. So first germ tube is formed, then it leads to the formation of aprosporium. Germ tube also perceives the host surface, and if it does not receive appropriate external stimuli, it remains undifferentiated. And when nutrients are exhausted, it will stop growing and dies. So if it is not perceived by the host properly, it will ultimately die. So appropriate physical and chemical signals such as host surface hardness, topography or hydrophobicity and plant signals, it leads to the germ tube extension and differentiation, right? So it requires certain kind of a perception by the host for its growth. So after germ tube, after some time, lead to the formation of a aprosporium. So aprosporium is a specialized cell which in many fungal plant pathogen, which is used to infect the host plant. It is flattened hyphal pressing organs from which minute infection peg goes and enters the host using turgor pressure and capable of punching the particular host, right? So this is how. So this is an example of a spore germination. This is through some high electron microscope kind of a thing, they can see this. So once the spore germinate, then germ tube grows and move towards the infection site. So you can see spores and its germ tube also. Again, it depends upon physical factors also, such as temperature, moisture, stomatal opening and condition. We'll study about the stomata, how they are entering through the stomata and other openings also, right? So factors which affect spore germination could be the moisture, could be the light, biological factors such as host nutrition, sugars and amino acids. Also the amount and duration of a particular factor determine the success or failure of a spore germination. So all these factors will ultimately decide whether the spores, they are going to germinate or they are not going to germinate, right? Then this is what are the pre-penetration processes? So in bacteria, they multiply in a moisture. So whenever some moisture is there, so they are going to multiply, or in some host executes, they are going to multiply, or they are going to increase their number, which is called as a pre-penetration process, right? Then pre-penetration process in a nematode. So in nematodes, the hatching of the eggs will take place, right? So nematodes, they have a number of steps, such as they lay eggs, then there's a juvenile stage, then there's a hatching juvenile, then there's a penetrating juvenile. So in different stages are there, right? So in a pre-penetration process in nematodes, they are going to hatch their eggs. Then penetration process. Again, it is direct penetration and indirect penetration. So fungi can penetrate either way. It could be direct penetration also. it could be indirect, whereas bacteria mostly enter through wounds and through the natural openings. And viruses and phytoplasma, they are transmitted by the vectors. They get entry through the vectors, right? We have studied a lot of vectors like Bemisia tabeca is insects, or even the nematodes act as a vector of the viruses. So they are taken inside by the vectors. So direct penetration, this is the example. They've given different examples for germ tubes, apple scab in Venturi and Nicolis. Then Aprosporium is formed in Calectotrichum species and M. grisa, right? Hostraria are being formed in powdery mildew or infection cushion, right? So germ tube is formed, Aprosporium is formed, then infection packs and the Hostrarium at the end, which will lead to the direct penetration. Then we know Hosteria. They are root-like structures that grows into or around another structures to absorb the nutrients, basically. So methods of a fungal penetration could be direct with the Hostoria. They can directly enter with the Hostoria. Then subcuticular, through the cuticle, they can enter, right? Then Aprosporium, direct with the Aprosporium and penetration pack formation. So these direct through the intracellular mycelium. So these are the different ways by which, or a direct penetration by which the fungus they get entry into the plant, right? Then the penetration by nematodes. So there is a second stage juvenile of nematode which actually penetrate through the root of the soybean. This is just example. So second juvenile stage in the nematode, they get entry to the plants. Like it could be the root nematode or a shoot nematode, different. So they can get entry directly. Then indirect penetration. So indirect means penetration through the natural opening. So we know that plants, they have several natural openings such as stomata, hydatodes or lenticels through which the pathogens can enter, right? So we know the stomata, they are pores on the surface of the leaves. Hydatodes is again a type of other pores which secrete water through this pores in the leaf margin. Then nectar through nectar glands, and lenticel, again it's a porous tissue which consists of cells with intercellular spaces. So through these, when there's a penetration through these means, it is called as an indirect penetration. So here we have a natural opening through stoma, lenticel or hydatodes. It can penetrate through stomata. So some fungal and bacterial pathogens, they penetrate through stomata, and it again depends upon the size of the stomata, number of the stomata, where it is located, and the time of opening and closing of the stomata also, right? The majority of the fungi, they again produces Aprosporium. Stomatal invaders are fungi which belong to the group Astilaginales, Iridiales, and Pernosporales. So these group of fungi, they mostly enter through the stomata. So this is again a figure which shows how it penetrates through the stomata. So we have a stomatal guard cells opening here. Again, iridospores, they germinate to form a germ tubes. Again, they form a Aprosporium. They enter through the substomatal vesicle, basically, right? And then they can enter into the mesophyll cell by the formation of the Hostorium. So this is how they are getting entry into the plant cells inside also. So stomatal penetration include Aprosporium, formation of Aprosporium, penetration peg, substomatal cavity, they move to the substomatal cavity, and then they form a infection hyphae. They can penetrate directly also, and sometimes they can form a Hostoria also, right? This is again entry through stomata. This is the enlarged view. You can see the spores, they are forming the germ tubes also, and they are forming the Hostoria-like structures also, right, inside the stomata. Then entry through hydrothodes is reported in bacteria Xanthomonas, nectrothodes in Erwinia amylovora, which cause fire blight of apple and pear, and lenticels, example is Erwinia amylovora again. So by different pathogens, bacteria and fungus, they can enter through different ways also. So hydrothodes, I told you earlier also, these are special glands or pores at the end of vascular tissues on the leaves through which water exudes and are natural openings for the black rot of the bacteria. Then we have a lenticels. Again, these are the small pores or openings in the outer skin of the plant. Again, these are the way by which gas exchange between the atmosphere and the plant cells, it takes place. So they can develop on any part of the plant, such as root, twigs, stems, or the trunks also. then we have a penetration through the wound. So this is very common way. If plants are wounded in any way, so they are open for infection to be caused by any bacterial pathogens also, fungal pathogens also, and viral pathogens also. Example, wounds can be caused by the hail, frost, or a heat scorching, or a insect feeding, right? So there are different ways by which plant can be wounded, right? The pathogen first multiply on wound in the presence of a moisture, and then it penetrates host cell, could be directly or could be hostoria, or it could release certain enzymes or the certain toxin also. So that way it can enter, right? So penetration through wounds also can occur in a plant, right? So wounds or natural cracks between the roots or a fungus, they kills and they macerate the cell ahead of its advance. So it keeps on moving through the wounds also, it can move inside, right? So this is about up till the penetration, how it is getting. So I'll stop here, I'll not go further. This next will go for an infection process and how it establish an infection with the host, right? All those things. What is the timeline of infection, and how what are the how what happens after the infection takes place. So that we are going to discuss in the next class. So if you have anything, you need a clarification of anything which you think you have not understood, you can ask your questions right now. Okay, so just go through these things, right? Just try to read what up till whatever taught you, just go through that. It is important, because agar aap aaj nahi padhoge, then you'll forget. I'm sure there are many new terms that we have used this time, and there are many new things which you have not studied earlier, right? So this is something new for you, many things are new for you, so you need to study, you need to go through these things. I'll also upload a PDF of this. This is a big PDF. I think it will be uploaded. There's a PDF file also of this lecture, so I'll try to upload that also. Just step-wise procedure. Just go through the things. What are polycyclic pathogens? What are monocyclic pathogens? What is primary inoculum? What is secondary inoculum? What is inoculum for that matter, right? So just try to understand these basic terms, and I'm sure if you'll just go through it once, you're going to understand. There's nothing very difficult, because I'm you see, I'm following a agrios. Agrios is a holy grail for a plant pathology, right? When we study plant pathology, we refer to agrios. GN Agrios, he's written a very good book, plant pathology, which include everything about plant pathogens, right? Everything from A to Z. So I'm following that book, and I'm trying to convert it into a very easy term, right? A very general term, so that you can understand. So मैंने बहुत सारी चीजों को modify करके और short form में लिखा है, so that you don't have to go through the complicated things. I just want to understand the basics. So right now what I want to understand is the basic, not going towards the other higher things or a higher terms. So मैंने बहुत basic करके मैं आपको पढ़ाने की कोशिश कर रही हूँ, but still I believe it is not tough. If you'll just go through the things, you'll understand it, right? But if you'll not just go through the things, you'll think that when exam will come, we'll study, then you're not going to understand them. You need to study them when I'm teaching you. And if you don't understand, then tomorrow we can discuss the things again, right? So please just go through the things once, when you after the class in the evening, or whenever you get a time, right? So then only you're going to understand the things that we are studying here, right? Okay, so I'll stop here, I'll not go it further. And in the meanwhile, I'll just give you one small assignment question also for today. Just do uh, after the class, or whenever you get time, or maybe right now if you have certain time, right? Just a moment. Okay, I've just assigned 2 small questions in the quiz. Just quickly, just do whatever you've understood in your own language. Just quickly within, we still have 3 minutes, right? And rest for attendance. So within 3 minutes, submit and come back. Students, I've uploaded a PDF file which is named as basics. So this all lecture for today and the next, which I'm going to teach you about infection also, is being included in this particular PDF file. Just try to go through this, right? Okay, so
Ma'am, I'm
yeah.
not able to submit my answers. So, is it okay if I try again in the evenings
Yeah,
and
you can
pay
do
the
it. No
submission?
issues. Yeah, no problem. You
Okay,
can do
thank
it, right? So please switch on your cameras for attendance. Adhish Mishra, Athoiba, Ayushi. Okay, Athoiba, you're
Present,
there.
ma'am.
Sam,
Present.
Present, ma'am.
Devanshi, yeah,
Present, ma'am.
Devanshi is there. Ishita, she's
Present,
there.
ma'am.
Janvi Singh, yeah, she's there. Kshitij, yes, Kshitij, are you there? Yes, Kshitij is there. Manaswani, yeah, Manaswani, I saw. Namrata Pal,
Present, ma'am.
Nandini Raj. Yeah, Namrata is there. Nandini. Nehal, Rupal,
Present, ma'am.
Present, ma'am.
Sahiba, Sanjana,
Present, ma'am.
Shakti, Shivani,
Present, ma'am.
Tanvi,
Present, ma'am.
Present, ma'am.
Tridev. Yeah, Tridev is there. Shakti is there. Yukta, Yuvraj. Vivek is not there.
Present, ma'am.
Present, ma'am.
Okay, Vivek is not there, right? Rest all are there and Vivek is not there. Okay. Okay, students. So we need to since you guys in March you are going for a project, so we have a February. So February is for exams also, test also, and for presentations also. So let me finish up this the second module also. So what I want is that we have a fifth module. I guess it's a type diseases, right? The diseases which talks about the diseases caused by particular pathogen in a plant. So that module I would like you all to give as a presentations. How and when? So I'll decide. We'll do group wise. Let's not, because there are 5 to 6 diseases, so we'll not be able to do individually. We'll divide again like last time we did one disease for a few people to make a presentation in a particular group. So maybe 3 or 5 group will make, and we'll divide students in the group. So you all can prepare presentations in a group for that. So basically not January, but February will do the presentation, and then maybe after 15th February somewhere we'll do the exam also, right? Class test also. So just keep preparing for your course side by side. Just try to go through the things alongside when we are studying them in the class, right? Okay, students. So I'll see you tomorrow again with the class. Thank you so much.
Thank you, ma'am.
Thank you, ma'am.